The objective for this experiment is to determine the effect of light distance on the rate of photosynthesis.
Photosynthesis is the process by which autotrophic organisms use light energy to make sugar and oxygen gas from carbon dioxide and water. Photosynthesis occurs by capturing energy from sunlight to make ATP and reduce NAD+ to NADPH and use the ATP and NADPH to synthesize organic molecule which is glucose from carbon dioxide. The rate of photosynthesis is the gross measure of the rate at which a plant captures radiant energy and fixes inorganic carbon compound. Leaf discs normally float in solution because of the presence of oxygen. However, when the discs are degassed, they become denser and therefore sinks. The solution of sodium bicarbonate acts as the sources of carbon for the process of photosynthesis to occur. As photosynthesis progresses, oxygen is released and the consistency of these discs changes, causing them to float. At the same time, cellular respiration is occurring and consuming oxygen. This makes the rate of the disc rising in any direct measure of photosynthesis occurring in the solutions. There are some procedures used in this experiment to make them occur more.
The hypothesis for this experiment is if the light distance is increased, the rate of photosynthesis is decreased due to the light intensity. Using a hole punch, make 10 leaf discs from the orthosipens terminus leaf. Then, the plunger have been removed from the strings and the cover have been removed from the tip. Put the leaf discs into the barrel of strings and tap them down to the tip, and we must be careful while replace the plunger into the string so not damage the leaf disc. In a 500 ml beaker, 0.5 grams of sodium bicarbonate have been mixed with 50 ml of tap water. Then, about 6 until 8 ml of bicarbonate solutions have been drowned into the strings. The leaf discs should float in the solutions. Next, the strings have been held with the tip up and expel the air by gently pushing on the plunger. And then, the tip of strings have been plucked lightly with our finger and gently pull the plunger, creating a slight vacuum. The vacuums have been held for a few seconds and then release the plunger, let it back. Then, repeat all the previous steps until all the leaf discs have been sunk to the bottom of the solution. When all the leaf discs have been settled to the bottom of the solutions, carefully remove the plunger and pour the disc and solution into the cup. They should settle to the bottom of the cup. The torchlight have been set up so that it's suspended about 3 cm above the beaker. Turn on the light and start a timer. Watch the leaf discs at the bottom of the cup and start record the observation, which is the floating of leaf discs every minute until all the discs are floating. Once finished, the light have been removed and cover the cup with aluminium foil condition for 15 minutes. After complete the 15 minutes, remove the aluminium foil and see the observations. Repeat all the previous steps, but this time set up the torchlight that is suspended about 10 cm above the beaker and record the observation. Based on the result, the rate of photosynthesis of floating discs with light distance 3 cm is greater than rate of photosynthesis of floating discs with light distance 10 cm due to light intensity. Increasing the light intensity increases the rate of photosynthesis. And then, based on the observation, the leaf disc will sink back to the bottom of cup after covered by aluminium foil for 15 minutes because the absence of light. Without light, photosynthesis cannot occur and there are no producing of oxygen gas that cause the leaf disc floating on the surface of solution. From this experiment, the suction can help the leaf discs to sink due to the air in the space between cells of leaf, which help them to collect carbon dioxide gas from their environment to use in photosynthesis. When a gentle vacuum was applied to the leaf discs in solutions, this air is forced out and replaced with solution, causing the leaf to sink. Then, if this experiment was conducted without baking soda, the leaf discs will keep floating in the solution because the leaf discs have air in the space between cells, which help them to collect carbon dioxide gas from their environment to use in photosynthesis. Baking soda provides carbon dioxide gas for leaf discs to photosynthesis. Then, when exposed to light, the leaf discs use carbon dioxide and water to produce oxygen and glucose. Oxygen released from the leaf form tiny bubbles that cause the leaf to float. Lastly, during the dark condition, the leaf disc will sink to the bottom of the cup due to the absence of light because there is no photosynthesis will occur, so no more oxygen gas will be produced. However, respiration continues in the dark, so the disc will use the accumulated oxygen gas. They will also produce carbon dioxide gas during respiration, but carbon dioxide dissolves into the surrounding water much more easily than oxygen gas does and isn't trapped in the interstitial space. The conclusion from this experiment is we can conclude that the rate of photosynthesis are affected by the distance of light.
If the light distance is increased, the rate of photosynthesis will decrease. That happens because of the decreasing of light intensity. Light intensity refers to the strength or amount of light produced by a specific lamp sources. Without enough light, a plant cannot photosynthesis very quickly, and the further of light move from the plant, the fewer oxygen bubbles are produced by photosynthesis. When the light intensity will decrease, the rate of bubble production also decrease, so rate of photosynthesis also slow and decrease. That's all from me. Thank you.